Good morning, everyone, and thank you for having me here today. I'm very excited to talk about soybean diseases and planting considerations. So as Ryan mentioned, my name is Horacio Lopez-Nicora. I'm the soybean pathologist and nematologist here at Ohio State. So an agenda always keeps me on track, especially when we're talking about disease that I know everybody is very excited to hear about and talk about. So today we're gonna just focus on SDS, other soil-borne pathogens, including the water moles, Phytophthora, Pythium, and then jump a little bit and learn what we're doing in terms of evaluating fungal pathogens in the battle for the belt. So with that, I appreciate the previous presentation. Plant pathology 101, the disease triangle, it's always something that we bring to any talk. So the susceptible host, in this case, now we're going to talk about soybean, a virulent pathogen. We're going to talk about some fungi and fungal-like organisms. The water moles are not true fungi. And then the conducive environmental conditions, which in this case, we can be responsible for modifying. So when we select different planting dates, we will encounter different conducive environmental conditions for different potential diseases. The main take-home message, as you probably all know, is that you need the three pieces here of this triangle for disease to happen. If we remove one of these, we will not have disease. No matter how much pathogen we have in that field, if the host is not susceptible, if we don't have the conducive environmental conditions, we will not have disease. And we actually use this concept, very basic it seems, to develop management strategies. Okay, so sudden death syndrome, SDS, we're pretty familiarized with this disease. It happens every year in Ohio, sometimes more, sometimes less. The fungus responsible for this is Fusarium virguliforme. This species is the most prevalent one in the United States. There are different species of Fusarium that can cause SDS. So we are very familiarized with this symptom here. This symptom is what we call an intervenial chlorosis. So the veins remain green. In between the veins, the plant starts showing a yellow color, and then it dies, creating a premature defoliation of the plant. However, the fungus is not in the leaf. The fungus is in the root system. It's a soil-borne pathogen. It inhabits and lives in the soil. It produces survival structures to overwinter in the soil. And when we plant soybean, and the population of this organism is in our soils and the conditions favorable, like cool soil temperatures, the fungus will infect as soon as the plant germinates. So the fungus is inside the plant right after planting. And that may sound counterintuitive because we normally see the disease very late in the season, at R4, R5, very late. However, the pathogen is already causing damage very early in the season. So the fungus is a root brother, and that's the first punch that will give to the soybean to reduce yield. It will start colonizing and feeding on the root system. Eventually, the fungus will produce toxins that will be translocated upwards and cause that symptoms that we see. But the symptoms are a result of a toxin that is being produced in the root system and translocated upwards. So essentially, the fungus is producing a herbicide in the root system, sending it upwards, displaying these symptoms that is essentially the leaf dying and premature defoliation. And that's the second punch that the soybean receives, and of course, further yield reduction. As the season develops, the fungus continue growing, and then when the soybean is harvested, the fungus will remain in the soil, increasing its population and awaiting for next season. In fields with conditions such as very wet and irrigated fields, compaction, and the presence of soybean cyst nematode, the severity of SDS is higher. It's more severe. So in fields where we see a lot of SDS, it's a good practice to get a soil sample and send it for nematode counts, especially soybean cyst nematode. Okay, that's a life cycle. This is a video I got from a field that I visit where the grower calls and say, "Hey, I have SDS," and we visit the field. We wanted to confirm that it's SDS. We will talk about how the symptoms that it's caused by the fungus Fusarium SDS can be also found that is caused by other pathogens, and we'll see how we can distinguish this. But everything that you see there that is yellowish brown, that's SDS. That's prematurely defoliated plant due to this pathogen. I mean, pretty large areas affected by this. All these beans were treated with a product for SDS. So what happened? We'll talk about that at the end of SDS. So this is what we're seeing. Yellowing at the beginning, in between those veins, became really evident where those veins remain green, and in between that, the tissue is dying, and of course that will cause premature defoliation. And we can see some of the pots there where they really need that green area to produce photosynthase. Those will definitely be damaged. I mentioned that there are other organisms that can cause exactly the same symptoms. So if I'm in my truck and I just stop next to the road and I see these symptoms, I can say SDS, right? I'm pretty sure. However, that is the big danger of doing that, of not getting down, pulling some plants, splitting some stem, and seeing if the pith of the stem is not brown, completely rotten and discolored, with this, brown area here in the pith of the center of the stem. That's caused by a different fungus, Cadophora aggregata, and the disease is brown stem rot. Why is this so important? Well, the resistance material that works for SDS will not work for this one. So you may be having a situation where you can say, but I planted a resistant variety to SDS, yet I still have SDS in my field. Well, maybe you have a different pathogen. Very important to get down, to pull some plants. If you don't know, send it to the lab. We'll do that for you for sure. A pathogen that we have been hearing a lot this past season is the red crown rot. Again, same type of symptomology. Just walking those fields and looking at this leaf, we can say, hey, we have SDS, not a big issue. We have it every year normally. Well, we may have a new disease in that field. And we really need to know that to be able to figure out the different management strategy. It's a disease that is in Illinois, Kentucky, and now last year reported in Indiana for the first time. So we should be careful, not maybe underestimating by saying, hey, we have a little bit of SDS in my field, but maybe actually having this new disease. So if you have a time, hit that QR code. We have an article in the newsletter, core newsletter, that can tell you a little bit more of how to diagnose this disease in the field and how to submit your samples to the lab so we can help you as well. So we're doing all that chain, we're splitting the stems. We can see that for SDS, clearly we have a white pith here. It's not brown, but the root system is this color because this pathogen is feeding on that root system. It's damaging the soybean plant by reducing rhizospheric area. We pull those plants very gently, and we can actually see the fungus growing on that stem, very bluish, greenish in color when the soil is very moist. We take that under a microscope, and we can actually see tons of those spores, and each one of those can germinate and cause an infection to your plant. And these can also survive in the soil. So you can see how easily we can start building up inoculum in our fields. We are receiving samples from growers. Last year, we received samples from different fields that show SDS symptoms. They will send a root system. Again, sending the leaf will absolutely tell us nothing. The fungus is not in the leaf. We cannot retrieve organisms from that leaf. This is a result of a toxin that the fungus in the root is producing. So we need the root system to be able to positively say, we have Fusarium that is causing SDS in the field. This year, we have funding to, again, process those samples. You can send it to the lab. We receive those samples, and we inform you that we positively either detected the fungus that causes SDS or a different one. Again, I invite you to hit the QR code. It's a folder that I prepared for you with a lot of information, fact sheets, submission forms, and how to prepare samples to send it to the lab, which is sometimes a very valid question. I want to submit a leaf that has a weird lesion. How do I do it? Well, we have indications of how to do that. We want to test for SDS or other related pathogen. How do I do that? How do I submit? We have that information in that folder. So I invite you to go and visit that, as well as soil sample, which we do a lot. This is what we got so far last year. And again, every single time we receive these samples, we get that metadata of when did you plant, what time, and the conditions at which these plants were planted at. So 17 counties in Ohio, we had samples that were tested positive for SDS. And I invite you to read a little bit about this project that a PhD student in my lab is working on, Jenna Moore. Essentially, what we're doing, several objectives, and everything is related to different planting dates and when these fields were planted and conditions at planting, but essentially growing these different Fusarium that we're retrieving from plants that are showing SDS. And so much diversity we're finding. Further on, testing these to different levels of fungicide. Are these isolates developing resistance to some fungicides that are working very efficiently to manage and control SDS? We're evaluating that in the lab. Do they behave differently in terms of how aggressive they are to soybean? Is there a region in Ohio that particularly is more aggressive than others? Those are all the questions that Jenna is asking, and I invite you to read that article that I posted there, which I think you will appreciate. Okay, so when we talk about management for SDS, we always think about, check your soybeans' nematode numbers. There is evidence of interactions between these two organisms. Reduce compaction, crop rotation, not with corn, it says, and that's because recent studies show that the fungus can colonize corn root, however, not cause problems to corn, fortunately. But the population builds up, and then when you put soybean again, like SDS can be more severe. Planting date is the one that I italicize here because that's the one that we're focusing, asking questions about. Planting in cooler soils, we know is more, it can enhance severity of SDS. So how can we play with that based on what we're learning from Battle for the Belt and the pathology study that we're doing? And then seed treatment, one that is very effective. And we'll talk about how to select effective seed treatments when we cover Phytophthora here in a minute. But, the fungus that causes SDS and soybeans is nematode, they're not the only pathogens out there, right? We have a soil that is, loaded with different organisms, beneficial and not. So Fusarium graminearum that we talk about with corn, the one that caused gibberella erod, is the same that can cause seedling rot of soybean. Rhizoctonia, one organism that we saw a lot this season for some reason, is another one that we see very often causing seedling blight. And these are all, like the fungus that causes SDS, true fungi that we will need to select a specific product, fungicide, for our seed treatment. Which brings to what happened with those organisms that are not true fungi, like the water moles, Phytophthora and Pythium. So here we have an example of using some products that are meant, in this case, Captan and Maxim, to control a true fungus like Fusarium graminearum. And we see that all these plants have been inoculated with Fusarium graminearum, except the control that was, we use water there. And then you see that we're using another product, Apron Max here, that is meant solely to be used for Phytophthora and Pythium and has absolutely no effect on true fungi. And the same thing we'll see that happened for products when we're trying to manage Phytophthora and Pythium. Selection of the product that is meant to control the target pathogen is key. And I'll share with you where we can find this information. So switching a little bit into the water molds, Phytophthora and Pythium are the common ones that we have in Ohio. They require sitting water. We need sitting water for these organisms to be severe and damaging. They can cause dumping off, either pre-emergent or post-emergent dumping off. Phytophthora, if we don't have good genetic resistance or partial resistance for the pathogen, we will experience also loss of adult plants when we have saturated soils during the season. The organism will overwinter as oospores. That's the structures that will allow both of these pathogens to survive for long periods of time, which means that once you have the problem, you're not going to get rid of it. We need to figure out a way to manage. And we will manage by alleviating saturated soils, having drained soils where we're planting, no seeding waters. They rely on seeding waters for infection. Seed treatment, selecting the correct seed treatment, as I showed in the previous picture, the ones that are meant to control water molds, and genetic resistance. Read the package. We have information on partial resistance that we can use when we're selecting the seed that we will plant in our field. You can see the different species behave very aggressive under different temperature conditions. And that temperature condition pretty much span through our window of planting and growing season. Just as an example, a publication that came out of Dr. Ann Doran's lab in 2020, soils were incubated at different temperatures. And by just changing the temperature at which they were incubated, they were able to retrieve, as you can see here, based on the color, different species of Pythium and Phytophthora and other organisms, water mold in this study. So again, we're thinking about planting dates. Are we planting in cooler soils, in warmer soils? Well, that will change the microbial composition for sure. Genetic, like I mentioned, is something that we need to keep an eye on all the time. Read that label. Do we have a rating for specific organisms? And what are the types of genes or multiple genes that we can use to manage Phytophthora in the field? I will share with you in the chat, but also if you went to that QR code that I share with you on the folder that I prepare, you will find a document and fact sheets where we have different products. So seed treatment efficacy trials, we call it. So what we do is plant pathologists across the Midwest, we get together at the beginning of the year normally, and we start sharing data from our different seed treatment trials. And we start rating products based on the different pathogens that we evaluated. So for example, we have 3 products here, active ingredients that work excellent for Phytophthora and Pythium. However, you can see that for the true fungi, they just really don't do a good job. It's not recommended at all. Same thing with the example that I previously showed, where we have products that are extremely good for the true fungi, but poor or not recommended when we use them for the water molds, for Phytophthora, for example. So you will find this information, and I know Ryan is also sharing that link in the chat. So check that out. Very useful when we're making decisions on what to apply. And I hope the question that you're having now is like, well, but you just say that we have different pathogens in the field, and you're showing me that I only have 3 active ingredients here for my Phytophthora groups, and then these other ones for my true fungi. Well, fortunately, we have some trade name products that will contain these different active ingredients. And that's something that we That's something that we need to pay attention to, right? What are the active ingredients that this product that I'm using as seed treatment have? And if I know the history of my field, then I can make a really good selection of what to use. Which led us to these final considerations for seed treatment. Do I know the history of my field? Do I know that I have, seedling diseases, especially in some areas that retain water more, or do I have a nematode at high population? I noticed in some years more severe symptoms of SDS. I'm planting earlier because I'm following some recommendation and also some windows where I actually can go and put the seed in the ground. But are the soil becoming cooler because of that? So those are all indications that maybe I need to protect my seed with a seed treatment on top of all the other integrated management that I'm doing. Cultural practices, draining my soil, selecting a good genetic materials to plant, etc. Stephanie presented a great information about cover crops, right? That green bridge that will just maybe allow for an increase in populations of organisms that are beneficial, but others that may not be. So all these considerations are important to take into account when we are deciding on what seed treatment we will use. And then finally, to wrap up here on the last minutes, I want to talk a little bit about the fungal pathogens that we are evaluating in the Battle for the Belt project that Laura and Osler are leading. And again, this is a massive work conducted by a PhD student in my lab, Jenna Moore, where we are, leveraging on these experimental designs, which is the Battle for the Belt. And that's a really neat thing here. It's not like we're just going to do some descriptive survey here. We actually have reps, we have blocks, we have an experimental designs where we are retrieving samples from 3 locations, 5 different planting days, 2 crops, and we are looking at the microbial community from these seedlings. So what Jenna is doing is essentially fulfilling 3 objectives. One is evaluating these environmental conditions, analyzing soils variables. So looking at the different parameters of soils, but also the microbial community of the soil, that soil that surrounds the seedling when it germinates. And then also evaluating what are the different organisms that she can retrieve from those seedlings. So we have different microbes surrounding the seedlings in that rhizosphere soil, but then she's also retrieving fungal organisms from those seedlings, cleaning them to make sure that we are not just identifying and evaluating contaminants, but only the ones that were able to infect the seedlings and we were able to retrieve it back. And that's how she does it. She surface sterilized these seedlings that are coming from different dates, and then she played it in different selective media. So we can pull the water mold, so we can pull the true fungus, the true fungi, and then she characterized them to genus and species level. Massive work. So far, we have 90 fungal isolates, 65 fungal isolates from soybean, 90 from corn. They have been also molecularly sequenced, so she can validate what she is looking under the microscope. 60 oomycetes, the water mold for soybean, same for corn, and the sequencing is just ongoing. And the next step is that we want to apply some metagenomic analysis on the soil and the tissue components. So much more data that we're gonna incorporate and get from these planting date experiments. With that, of course, I want to thank my lab members. Without them, I will not be here presenting this, and I want to also thank a lot of great people that we work with in the different research stations. Without them, our program cannot produce the results that I'm showing. So I thank you for your attention. I think I have 2 or 3 minutes for questions, Ryan.